Well, good morning, everyone, at least here in the United States. My name is Mark Shelhamer. I'm the MC for today's event. I direct the Human Spaceflight Lab here at Johns Hopkins University at the School of Medicine, and also a program we call Bioastronautics at Hopkins that aims to promote human spaceflight not only here at the university, but other universities and institutions as well. And it's part of a larger umbrella group at Hopkins called Space at Hopkins. Our efforts here on the Symposium are supported by the Office of Research and Translation in the School of Engineering. Today's event is run by our partners, Hopkins at Home. They provide video and online promotion and production. This is the 7th of our online events. The first was a larger event in February of 2021. Since then, we've had 5 of these mini symposia on the topics of systems medicine, statistics, surgery, space radiation, and persons with disabilities flying into space. Today's topic is on omics and genomics specifically. We'll have presentations from 2 experts in this field, comments from a third speaker who is so far not deeply involved with human spaceflight research in this area, then a discussion between them and me, and finally questions from the audience. You can submit written questions online during the presentations, put them in the chat box on the Zoom software platform. Questions will be read by Malika Sarma to the panel when we get to that point. Malika, formerly my postdoc, now a faculty member at the University of Pennsylvania. Our timing is flexible based on the discussions among our panel and the questions, but we will end in 2 hours at the latest. So today we're going to talk about omics. That's a vague and general term that encompasses a lot of things: genomics, epigenetics, proteomics, metabolomics, and others. From the online Britannica Encyclopedia, I found this quote: "In biology, the word omics refers to the sum of constituents within a cell. The omics sciences share the overarching aim of identifying, describing, and quantifying the biomolecules and molecular processes that contribute to the form and function of cells and tissues." So basically, it's the study of genetic information and its processing by the body to create proteins and drive metabolic processes, and in our case, changes in these things during the course of spaceflight. So this is important for a number of reasons, among them the potential to implement precision countermeasures, kind of like precision medicine on Earth, for people in space. The twin study of a few years ago was NASA's first major foray into omics investigations in human spaceflight. Chris Mason, one of our speakers today, was one of the principal investigators on that large project. This took advantage of the fact that Scott Kelly, who was to be in space for almost a year, had a twin brother, Mark Kelly, who was a retired astronaut. Hence, this was a natural control population of N equals 1, and a great opportunity to look at nature versus nurture in terms of the adaptation to spaceflight. This project ramped up very quickly. Proposals were solicited in July 2013. Grants were awarded in March 2014, and the project flew from March 2015 to March 2016. We're not going to talk about the twin study exclusively or in great detail today, but it does serve as some of the backdrop for what we will be talking about. That study was very enlightening because of not only the range of science involved, but also some of the logistical concerns that had to be addressed. For example, having to draw and preserve biosamples in space until they could be returned to Earth for analysis, and the provision of genetic counseling to the test subjects, the Kelly twins, and the recognition that genetic insights pertain not just to the specific subjects who have agreed to participate, but potentially to their entire family. So there are many ethical issues involved in that just started to get NASA into the business of omics type research in space. And of course, NASA is not the only player in this area now that there are commercial space flights going on and experiments in science being conducted on commercial space flight. But let me go back to that story. And one of the things that came up in the context of the twin study was our becoming familiar with GINA, GINA, the Genetic Information Non-Discrimination Act of 2008, which prohibits genetic information discrimination in employment. In other words, even if genetic screening were to tell us that someone is unusually susceptible, for example, to getting cancer from radiation exposure in space, that information cannot be used to prevent that person from becoming an astronaut. So as we like to say in this field, our job in human spaceflight research is not to say who can and cannot fly, but to enable everyone to fly who wants to fly, both government and commercial. So within that context, the kind of information that we get from omics studies provides information to temper expectations, to design personalized countermeasures and interventions. So there are two aspects to this work as applied to spaceflight. First, basic research and a building of a database in response to spaceflight. And second, the development of personalized countermeasures, including possibly pre-flight activities for the adverse effects of spaceflight. But I want to emphasize here, partly because I don't work in the omics area, I work in other areas of human spaceflight research. So I want to emphasize that omics is not the only thing going on in biomedical research, let alone space biomedical research. Omics work is useful to the extent that it can provide insights into the things that really concern us about humans in space, the ability of humans to fly safely and productively. There are, of course, fundamental scientific insights that might be gained from this type of work, the biological response to multiple simultaneous stressors, for example, and I'm a big fan of basic science in space. However, I will posit that the true value of this work will be in providing insights and interventions to maintain health and performance. So I'll leave that statement hanging there for a little while for our speakers to contemplate while I introduce them very briefly. Now, my introduction here, I'm going to introduce all three of our speakers, two of our main speakers and one kind of a commentator on the two main speakers. I have to keep these brief because if I were going to go into the details of all of their professional accomplishments, we would have very little time left for presentations and discussions. So suffice it to say, you can find more information on the website for the symposium announcement. All very accomplished in the field, all have wonderful things to say. I'm looking forward to hearing from them. Our first speaker is going to be Michael Schmidt. He's the CEO and Chief Scientist at Sovaris Aerospace. Next will be Chris Mason, Professor of Genomics at Weill Cornell Medicine and Director of WorldQuant Initiative for Quantitative Prediction. And Chris Bradburne, the commentator, a member of the senior professional staff at the Johns Hopkins University Applied Physics Laboratory with the faculty appointment at the School of Medicine, like I have. So again, I will remind you that as the talks go on, please enter your questions into the chat box. And with that, I'm going to turn it over to our first speaker this morning, Michael Schmidt.
Great. Thank you, Dr. Shelhamer. And I wanted to thank you and your team and also Johns Hopkins for having us a part of this symposium. Obviously, this is a subject that we're quite excited about. And in terms of the growth of the space industry, right now, it's a pretty extraordinary time in history. And one of the requests that was made was that we get on the same page with regard to what is a gene. And now I'm finding that my slides advancing. So there we go. So just so that everybody is on the same page with regard to what a gene is, it would be worth summarizing this just briefly. And essentially, you look inside the cell, and in the nucleus here is a chromosome. There's a whole set of chromosomes that everybody shares. And then within the chromosome, there is the DNA, and then the segments of DNA, and I'll give the simplified version, is essentially what codes for a protein. And so the typical DNA markers that you hear about, the A, T, G, and C, they line up here. And together, that gets translated into what's called RNA. And then from the RNA, that gets read into something called an amino acid. And from this pause here, I'm having to apologize. The advancing of the slides is going a little slow in Zoom right now. It forms an amino acid, and these amino acids come together to form what's called a protein. And then the protein is really what has its effect on human physiology. So essentially, the components of our inheritance and the things that instruct us how to make things, in particular proteins, are really what the genes are all about. And there's over 20,000 of these genes, different kinds of RNA, many different kinds of proteins. But essentially what's important to understand about this is that these can all be measured. So when we talk about the concept of omics that Dr. Shelhamer spoke about, we're really talking about the ability to measure at each stage here. So as we try to describe the spaceflight environment, we can now measure all kinds of RNA, all the amino acids, thousands of proteins, of course, all of the genes. And now we can start to explain what's happening in space. But more critically, as Dr. Shelhamer said, we can start to talk about development of countermeasures. And so the one I want to pay a little bit of particular attention to here is what we call ApoE4. We'll get to that in a moment. But what I also want to say is the two talks that we have here, so Dr. Mason and I and our groups work very closely together on all these omics efforts in human spaceflight. And so what we'll try to do here together is in my talk, I'm going to speak about what are the targeted parts of the omics, where we can actually start to think about the effects of particular genes and the countermeasures that we might develop and apply in space. And then Dr. Mason is going to talk about the multi-omics concepts and in practice. So let's just think about a quick concept so we are understanding things together, and that is the term genomics versus genetics. So quickly, genomics is really the study of the entire genome and all of the interactions that take place within that genome, whereas the field of genetics is an older discipline, and that's really the study of individual genes and their inheritance patterns. And so today we're really talking about genomics, but there will be aspects of genetics here when we talk about individual genes and risk that are going to become quite important. So how we think about genes. Usually the general public thinks about the genome in terms of heredity. We're inheriting genes or alleles from each parent, and then we know in a general way that genes influence a lot of things. But from the standpoint of the specialists in this area, we're really thinking about genome, epigenome, transcriptome, and all of those components that I just shared. And we have to measure and characterize these and then assign biological meaning to each one of these. So as we go forward in this discussion today, I'd like you to think about a few basic questions. And one of them is, how do our inherited genes, and in particular, we'll talk about something called single nucleotide polymorphisms. You can think of those as maybe a mutation or a variant that impacts the function of a gene. So how do these inherited characteristics impact individual risk in space? The second question I'd like you to think about is, how does the space environment itself affect that gene expression? So genes have a capacity to do specific things, but under various influences, that can really change, and that term we use is gene expression. And so that is easily measured by something called RNA. And Chris is going to talk about that. I'll talk about that somewhat as well. But really one of the critical features, to get back to Mark Shelhamer's point, what are the factors that impact gene expression? In other words, beyond the space environment, what are all these intersecting factors? And then what can we do about it? Because really the critical point is first understanding these patterns, but then we want to develop solutions. And one of the things that these multi-omics experiments do is they can point us in new directions for new hypotheses that can then lead us to new experimental designs and then new countermeasures for spaceflight. And we view this as being able to accelerate the path of discovery in space. So let's use a bit of a metaphor here. Some of you who may have been in Tokyo understand trying to navigate the Tokyo subway route map. And this is a bit what it looks like. And if you don't have a map and you don't know what all the nodes do, you really can't get anywhere. We have a bit of the same problem in human metabolism, and we have our own map, and it comes out of the KEGG pathway map. And but our challenge is high dimensionality or high complexity. But we have to capture this complexity and start to reduce it into countermeasures. So we do this by characterizing aspects of it. And this is where all of these measurements, like the genomics, comes in. We start to characterize what's in here. But overall, what the multi-omics has done is it's allowed us to essentially, rather than just characterize specific boxes or what's in specific pathways, really analyze everything in the map. And so where that allows us to go then is we can look at what's coming into the system in terms of food, nutritions, vitamins, minerals, drugs, environmental exposures. We can now map the pathways and start to describe that meaning, and then we can talk about how it affects our physiology, behavior, performance, recovery, and our health and our safety. So that's really gets us to the primary questions then of we need to then take all this molecular data and reduce it into something that we can explain clearly. And in the end, this is where we're trying to go. We're trying to take as much information, and this includes molecular data, and to Mark's point, physiologic data, all the other kinds of measures. We want to understand beforehand how we can impact optimizing the astronaut for spaceflight, but we know that space has an impact on their physiology, and they respond differently. And some are actually poor adapters to space or poor responders to our countermeasures. Some are very good adapters. We want to try and move everyone up to the optimal adapter in the best sense possible. So I'd like to just very quickly, in kind of a rapid fire sense, go through about 5 main points to illustrate how to think about genomics in space. We'll use the Alzheimer's gene and how that may impact our brain in space. We'll talk about genes that might impact depression and anxiety in space. How do we pick the right drugs using genomics? And then the gut has genes too. So we're going to go gene hunting in the microbiome. And this is some findings that Chris Mason and I have made from the NASA twin study that's very interesting. Then how do we protect the DNA in space? And then Chris will get more to this. I'll leave that alone. So let's go quickly to the Alzheimer's gene. And these are the single nucleotide polymorphisms that I talked about. So a genetic variant in one particular gene. So we'll talk about ApoE. How does that affect the cognitive risk in space? And is there a protective genotype for this gene that could influence some people responding better in space than others? So apologizing for the graph, but this is really to make a point that there's severe degeneration, mild, and then the optimal healthy condition. So in one type of the genotype, and this is on Earth, you can see here that those who have this ApoE2 fare much better in terms of cognitive decline, and it trends much later in life. But then we have the extreme other end, which is the ApoE4, and you can see that this trends much more rapidly toward cognitive decline. This presenilin is just to show another gene significantly affects cognitive decline, just to give an illustration of the importance of these genes. So just to illustrate here quickly, genes actually can change something like even the shape of the protein. So I just use this as a little bit of an illustration. The ApoE4 gene actually changes the shape of the protein so much that its function changes. So the question is an ApoE2 genotype protective in space? And is an ApoE4 genotype at risk in space? And can we find a countermeasure for that? So let's look at some Earth-based studies. So for example, this is looking at chemotherapy. The question that's being asked is, in those receiving chemotherapy, do the ApoE4 genotypes respond the same or more poorly to chemotherapy? And the answer in several studies is that yes, if you have the ApoE4 genotype, what we're calling the risk genotype, that your risk to cognitive decline in chemotherapy is greater. So it essentially moves you even further back on the curve that I was just showing. And I'm going to try to save time here, so I'll skip going over each of the studies in breast cancer. We have studies in testicular cancer, and typically visual memory, spatial memory, mental state exam, all of these things do change, especially in the ApoE4 genotypes. And they last for many years. So then let's look at the question of radiation, because chemotherapy doesn't happen in space. So what happens with radiation? In cases of cranial irradiation, significant cognitive decline happens. Now, there haven't been a lot of studies comparing the ApoE2 and the ApoE4, but those are underway. But at the moment, what we do know is that it changes substantially with cranial irradiation. So the cognitive decline with radiation exposure is significant. And now it raises the question, and we'll skip this just for expediency. So if we were on a mission to the moon or Mars, do these ApoE genotypes have any impact? In other words, if I'm flying to space with an ApoE44 genotype, do I have a greater risk of cognitive decline than someone with an ApoE22 genotype? And this is an unanswered question in human spaceflight, but it's an example of how we have to think about understanding genotypes and the kinds of exposures into which we're entering. And then the other question is, can we move that risk? So if you have an ApoE44 genotype, could we move it in this direction? And could we even move it further and more proximal to the ApoE22 genotype? So these are the questions of countermeasures, and how do we pursue those? So I'll give you one example, and this is a study on Earth, but it's looking at the factors that influence the development of cognitive decline. What they show that those who are in the highest intake of phosphatidylcholine, this is a nutrient found heavily in eggs, there was a 28% lower risk of dementia, and each 50 milligram day intake of phosphatidylcholine was associated with a 10% lower risk of dementia. But the promising part of this was it was even in the ApoE44 carriers. So now we can start to think about genes in terms of risk and the different genotypes, but also in terms of countermeasures. And now this sets us on a course of pursuing the kind of countermeasures that could affect positively your genotype in space. So how should we think about this? Is ApoE4 a risk in space? Is ApoE2 protective in space? Should we screen all astronauts today? Because we aren't doing that, at least not that's outside of their medical records that can be disclosed. Should they all be on phosphatidylcholine at this moment? But are there other countermeasures? And then what should a flight surgeon do in clinical care today? So these are all the questions we're wrestling with, but it's a way to think about genotypes in space. So let's think then very quickly about another kind of genotype. We all know what tryptophan, the amino acid, is. We convert it into serotonin and melatonin. Obviously, we know serotonin is associated with mood and melatonin with sleep. But tryptophan goes another pathway that's excitatory. And this can have very neurotoxic effects, this compound here. Well, it turns out that cortisol affects the genes or the enzyme that push things this direction. And cytokines, and Chris will talk about this later from our work with the twin study and other studies in space, that inflammatory cytokines can change in space. So we may have the conditions in space that accelerate the development of quinolinic acid, and we're looking at that right now. But if we understand all of these processes, we can realize that if we give tryptophan, we continue to funnel this pathway. But if we were to give the astronauts 5-hydroxytryptophan, it's not acted upon by these genes. And so we can fuel the serotonin and melatonin pathways by using this instead. So this is the reason we need to understand genes and genotypes, but also the proteins and metabolites that go along with them. Then we have the question of using genomics to pick the right drugs in space. And so there's a whole class of enzymes we talk about that are called CYP450, and genes control all of this. So I use drugs that everybody knows about, codeine, tramadol, and morphine. So if we pick this one genotype, we ask the question about its conversion to morphine. We have a class called poor metabolizers, and they convert about 10% of their codeine to morphine, which isn't very good and it's not very useful. We have the ultra rapid metabolizers that convert 40 to 50%. These are a risk genotype, and these are the symptoms. So imagine somebody going on a spacewalk who's in pain or who has upper respiratory infection, and they're given codeine to modulate their symptoms. But they're in this their classification, they may have any one of these symptoms on a spacewalk. So the flight surgeon would say, we can't use this because it's not effective, but we cannot use this in these astronauts because it's dangerous. So actually, we can only use these drugs in the normal metabolizers and the intermediate metabolites. And tramadol has its own risks. And so this now gives us a window into understanding a genotype. So this is some work of ours looking at, and these are the genes across the top. And you can see the red would be the rapid metabolizers. And so what I want to share here, this is actually a cohort of ours where we know the genotypes, but it's representative of what we would see in an astronaut population. And I'll look at the two because there's some relevance here. So in this classification, what we would do here in the area of these drugs, which are mostly antidepressants as a class, you can see in all the red, if they had this ultra-rapid genotype, we would select an alternative drug or monitor very closely in almost every case, and in some cases, specifically select an alternative drug. So we can build profiles lists like this for every astronaut based on genotype. And so I'm going to go through, skip these in the interest of time. Let's go back to one with caffeine just for a moment. So caffeine is metabolized through a different enzyme in the cytochrome area, 1A2. And just very quickly, about half the people are poor metabolizers and about half are the rapid metabolizers of caffeine. We think, well, caffeine's really not a problem in space. But if you look at this study, those who were the slow metabolizers of caffeine and who consumed caffeine at a high level, they were 2.5 to almost 3 times more likely to have all problems associated with kidney function and blood pressure. So we might say to an astronaut in space, if you're a 1A2 slow metabolizer, we don't think caffeine at any levels at all, certainly not high levels, are safe for the astronaut. So already we have 2 genotypes that we can make a determination of usefulness for the astronaut. And there are many, many others that affect many other drugs. Presently, there's about 500 drugs that have been given specific recommendations based on genotype. So that field is rapidly expanding, and it's important that it be expanded into human spaceflight. So now let's look at genes in the microbiome. We call this gene hunting in the microbiome because when we think of the genome, we're always thinking about the human genome, but we have a very large genome in the gut. And so this is some of the work that the team and the NASA Twin study has done. And now I'm going to talk about the work that Chris Mason and our teams have done together here. But we looked at the untargeted metabolome, and we found a signal here that was fairly notable. In other words, there's a compound here that's elevating in space called paracresol. So what this is going at the genome from in the opposite direction and saying, we found a metabolite, a small molecule, which you can see here. It's a very simple molecule. That's essentially telling us there is some activity in the genome bringing this about. And we know that this activity is in the microbiome. So now we're looking for microbes in the gut that are producing paracresol, because paracresol has several different functions that are important in astronauts. They're certainly important on Earth. And so I think this is the first finding of this in space that we're aware of. But essentially, dietary tyrosine is the precursor. So tyrosine is ingested, the gut bacteria convert that. One thing we know is it alters drug metabolism. The other thing that we now know, it influences some of the compounds associated with Parkinson's, liver metabolism, and even bone formation. And we also know that the butyrate producers are a class of microbes in the gut that make very important protective compounds on Earth and in space. And this paracresol inhibits those, but it stimulates the clostridial organisms, which we know are paracresol producers. So this is the adverse side of paracresol in a space environment. But we've also looked at the countermeasures, and what we know from Earth-based research, that there's a range of resistant starch and different fibers, that if you increase those in the diet, you lower paracresol. And so this can have the attendant positive effects on all of these different components that I mentioned that were the adverse effects of having elevated paracresol. So already we have from the genomic data and the untargeted metabolomic data, we have found a new marker that could become a new risk in space. It needs additional research, of course, but already we have countermeasure concepts that we can use to modify that. So the question might come in, and we'll just go back for a moment. Well, if dietary tyrosine is the substrate, the precursor to make all of this stuff, why don't we just restrict this? Problem is tyrosine is needed for all kinds of compounds in the body, in the brain in particular, but all kinds of protein. Astronauts tend to have low intakes of protein anyway, so we have to rule reducing tyrosine out because it has other risks. That's why these other concepts of countermeasures start to become more important. And we can look at these on Earth, we can look at the microbes in the gut on Earth, and we can start to build profiles for astronauts and then monitor this in space. So let's go to a last concept here, a last primary concept, and that is, well, omics and genomics in space is important, but how do we protect that DNA? I want to speak just briefly about DNA repair. So DNA repair takes place with a range of enzymes. We won't go into all of these, but APE, DNA ligase, topoisomerase. And then they all involve ATP, but one of the critical things about these enzymes is they are all magnesium dependent. If you remove magnesium from the environment, these enzymes of DNA repair really start to suffer significantly, and so their capability to repair DNA is diminished. So there have been several studies. Right now, there's 2 studies at least that have looked at magnesium losses in space, because we know muscle is a reservoir of magnesium, bone is also, but when you lose muscle and bone in space, magnesium is lost with it and can be eliminated in the urine. So the early studies showed there was considerable magnesium loss. More recent studies have raised questions of whether that's so, but I will say the recent studies involve astronauts receiving about 420 milligrams a day, so they are being supplemented. Question is that enough to protect DNA in space? We know from historical data in certain cultures that up to 700 milligrams are ingested every day. So we may need much more magnesium on a daily basis to protect DNA repair. And so these are the questions we're asking now. How do we maintain DNA integrity in space? And what are the different inputs? So let's go to 2 others, and then we'll finish up. And one is, so space radiation triggers a process called PARP. And basically, it says we need a protein to come and stick onto the DNA injury so that the repair enzymes can come in. But what that does is it steals from another molecular pool. It's called the NAD pool. It uses about 200 molecules of NAD to make one PARP molecule to fix just one DNA strand break. And so what happens? We start to deplete the NAD pool. And why do we care about that? So going back to our metabolic map, and let's just go to energy metabolism for a moment here. Just look here, here there's 7 steps where NAD is needed, but you can see NAD here, NAD here, NAD here, and 2 NAD over here. This is the foundation of energy metabolism. And this is just a handful of the 400 enzymatic processes that involves this NAD. So the question is, as we deplete NAD to repair DNA from radiation injury, how are we impacting these other processes? And do we need to replete the NAD pool with either NAD itself or precursors like nicotinamide riboside? But this is again where we think about moving immediately from understanding genomics into the countermeasure area. And then as a final quick note, and then we'll have one set here to go through, B12 folate and betaine, they provide methyl groups. So to actually make the basis of genomic structure, if you don't have enough methyl groups, the DNA incorporates the wrong nucleotide into its structure, and it's read immediately as a mutational event. So think about that. Going into space, if you're B12 folate or betaine deficient, you could actually be entering space with mutational processes going on. When you add radiation to that, that is a geometrically amplifying effect. And so we need to make sure that all of these compounds are in sufficient levels to maintain DNA stability and to maintain it for certain in the presence of space radiation. So just quickly then, one of the ways to reduce this complexity is the development of an astronaut digital twin. And those of you in engineering know the digital twin all too well. We go from an aircraft engine to a digital engine, and now we can build models to test all kinds of temperature and strain, altitude, etc. and use the model to now understand how to best prepare for the use of the actual system. And now we're developing an astronaut digital twin that basically is able to build a digital mirror of an astronaut and start asking those questions in the digital environment. And so as a final point, I just want to bring up this slide because it's really the question. Those are examples of going from genomics to targeted genomics to asking questions about countermeasures. But I want to raise this as a kind of a why we need genomics question. And some of this data comes from Lori Snyder, who ran the astronaut exercise program in the space station for many years. But this is looking at different loading paradigms of exercise. And each one of these dots represents either the IRED or the ARED technology that's being used for exercise. And it's looking at different responses, knee strength, endurance, knee and ankle, etc. But looking at each one of these as an individual astronaut, the thing that really stands out is how different they are. These are really large error bars and considerable difference between astronauts. So you would say some of these are very good responders and some of these are poor responders. So how do we understand why this is a poor responder and why that's an optimal responder? And the conversations that Lori and I have had has been simply, we need to get down to the molecular level, further deeply into the molecular level, very much into the genotypes of these individuals and into the RNA transcriptome as well, to try to understand at a molecular level why these are so different. And if there is a genetic risk to being a poor responder, can we find countermeasures to tailor it to that individual? So this is a classic example of what happens in space. All of these different types of response. And that is where the field of genomics, in part, other fields as well, but since we're talking about genomics, so that is where this is going to help us start to answer these kinds of questions of why all the individual variability. So let's just go quickly then to the spaceomics question. So earlier you saw this map, and we talked about these targeted ways of doing it, which are useful but not comprehensive. So this is where the multi-omic space comes in, and we mentioned each of these subgroups. And a lot of work has been done now with the twin study, Inspiration, Polaris, Axiom, etc. And what I really want to do is, as I said, Chris Mason and I and many others are working on these multi-omics problems in space. And I want to take this opportunity to thank all of you for listening and really open this up, because Chris now is going to jump in here and really explore the multi-omics landscape with regard to NASA, commercial space, and other spaceflight applications. So I want to thank all of you for your time, and I'll turn it back to you, Dr. Shelhamer.
All right. Thank you, Dr. Schmidt. That was wonderful. Really lays the groundwork. I am full of questions, but I'm going to keep it. I'm going to keep this brief and just ask you a couple before we turn it over to Chris Mason for his presentation. So it occurs to me that one thing we didn't do that I should have done perhaps in my introduction is to remind people of the risks, the hazards to humans in space, at least as they're commonly delineated: altered gravity, isolation and confinement, distance from Earth, which is remoteness but enhances crew autonomy, which has psychological aspects in addition to others. Enhanced radiation, which, Michael, you talked about a number of times, and a closed and potentially hazardous environment, toxic substances in the environment, the spacecraft environment, potentially, not to mention a planetary environment, the soil, the regolith on the moon, the soil on Mars. And you've been talking about things that are modulated by gene expression. To what extent do we see gene expression change over the course of spaceflight? Because so far, you've emphasized in your talk, it seems to me, a lot of the things that you could learn pre-flight as a baseline of this particular type of person might need this type of intervention, and that's all great. But recognizing that it's important to recognize that within the context of the specific set of spaceflight stressors, which I just delineated, and the fact that they're all going on at the same time, and that we send astronauts, certainly professional astronauts, into space to actually do things, to actually to get work done, to be productive. So taking into account the specific stressors, the specific tasks that they might be called upon to perform, including working at a very high cognitive level, which you talked about to some extent, and incorporating the natural human variability from one person to the other. I think that helps people understand that the challenges that we face here. But let me get back to my question. If you see changes in gene expression, for example, and various manifestations of that in space, they could be, do we know the extent to which they are appropriately adaptive because they're helping people adapt to the stressors of spaceflight versus the extent to which these changes in gene expression might simply be adverse consequences of the spaceflight environment acting directly on the genetic processes, the genome, the omics processes. So hopefully you understand what I'm getting at in my question. It could be that if you see changes, it could be the fact that gene expression is changing due to radiation because we need DNA repair and we need other things to respond to radiation, but it could be that they're seeing changes due to detrimental effects of radiation directly on the gene expression pathways. Can you make sense of that question and say anything
Yeah,
about it?
yeah, no, that's a really helpful question to ask, Mark. And so, one of the things that comes out of the transcriptome work, and I would say first that is such a crucial type of measurement is the transcriptome, and Chris will talk more about that. In the twin study, there were something on the order of 7,000 genes that were significantly altered based on the gene expression in space. And so those are loosely sometimes referred to as space genes. But that's certainly a function of duration. And obviously, since distance is pretty fixed at the space station, but it's very critical to understand that, yes, some of those may be adaptive, some of those may be positively adaptive. And one of the reasons that it's so important to measure the proteome, the metabolome, et cetera, as well, is to understand, okay, if that's a positive adaptation, and I pulled this slide back up to address that. If it's a positive adaptation, then we wouldn't necessarily want to develop a countermeasure that would suppress that. So for example, here, we're using PARP. And as I said, PARP gets recruited to the site of DNA injury, and then it immediately starts to recruit the NAD pool. Well, PARP inhibitors are being used in cancer therapy because PARP can be a problem in certain cancers. But in space, our immediate assumption might be, well, it's protective because it's dealing with some measure of DNA injury. So we wouldn't want to inhibit PARP in space. But we do want to look downstream at what are the other effects of upregulated PARP activation. And this would be one of them, where it's now stealing hundreds upon thousands of molecules from the NAD pool. And that's where I wanted to share this concept. So now it's robbing from other parts of these pathways where we say, okay, we don't want to inhibit PARP, but we want to understand the molecular network sufficiently that we can support this part of as a countermeasure of repleting the NAD pool. That could be with NAD injections, or it could be with NAD precursors orally. So that would be one critical feature, trying to understand if it's adaptive or if it's maladaptive, or at what point does the adaptive become maladaptive, and that's all typically temporally related. But I think one of the key things is we want to try and understand this as much as we can on Earth, so that we bring them to a very strong level of self-regulation before they go into space, and then understand what is changing in space and how do we reduce our countermeasure suites to most comprehensive, but also those things that are most significantly impacting the individual. And I do want to say individual. There was a recent study that came out on CO2 levels in space. And the thing about that finding was, and at a certain level, it was suggesting that high CO2 might be beneficial. But if you look at the standard deviations, right, the error bars, it was still quite detrimental to some. And that's where I think the individual personalized part of this becomes important. And then I'll just say one last thing and wait for your thoughts. And that is, we need to be measuring the right tissue to ask the right question. So for drug metabolizing enzyme data from our different studies in the transcriptome, but we have white blood cells largely, and some epithelial cells and things like that. Well, most of the drug metabolizing activity takes place in the liver, some in the kidneys, some in the testes and places like that, but it's mostly in the liver. So the question is, if we have white blood cells and we have a transcriptome for drug metabolizing enzymes, does that actually tell us what the liver is doing? And I would say the answer is no. And so we do want to have the right tissue when we're asking and answering these questions, but it's sometimes hard to get the right tissue. But that's part of the complexity. But I'll turn that back over to you.
Yeah, okay, that's good. So I'm gonna do something that you're gonna find very annoying and maybe a little bit unfair. I'm gonna ask you a follow-up question, but I'm not gonna give you a chance to answer it because I want to turn it over to Chris Mason, because it's a question that is applicable to everyone on the panel today. And I want to, after Chris Bradburne has had time to speak, or maybe he'll talk about this as well, I want everybody to engage in this question. But you kind of set me up for it, because with all of the things going on, all of the multiple interacting stressors that are involved in spaceflight, the well-known and very pernicious range of human variability, and the fact that these are all extremely complicated metabolic net and gene expression networks, is it really possible to say anything of a specific insight? I mean, at some point, when do you say, well, this is really way too complicated, and if we pretended we knew the answer to this and targeted this specific pathway or that specific precursor, wow, we could be doing more harm than good because we just don't understand it. Or we could be doing more harm than good in half of the people, but not in the other half of the people. So I'm gonna put that out there for people to think about, because it's the complexity of the whole thing. I think, Michael, when you were wrapping up your answer, you were getting at what I think might be some of the answer, which is, well, at the least, let's make sure that these expression networks and metabolic pathways are functioning at a very high level. Maybe they just have to go about their business on their own, and we still have a lot to learn about them, but let's at least not inhibit their adaptive capability. Now, I'm putting some words into your mouth there, but dwell on that for a little while. And now I'm going to turn it over to Chris Mason.
All right. Well, thanks so much. And that's a great question. Actually links towards some of the papers we just published, some of them today. So actually, I'll get to this during the talk and then put a link in the chat. But thanks again to Dr. Shelhamer and the organizers, and really for having me here. It's a pleasure. Many old and new friends on this call, so really happy to be here. I will try and keep it to, I think it's 20 minutes, so I'm going to jump right in, share my screen. Okay, great. So there's a lot of work I'm going to show you again in a kind of quick preview from some things that's been published, and then a lot of work we've done recently with some of the commercial and civilian crews, and then a little bit of looking ahead, a bit of a philosophical ending at the end. So we'll start with something, a reason why we're all here is thinking a lot about how do we get to the moon, eventually then to Mars. And this is one of my favorite renderings that NASA made of what it will take to get there. But of course, the big question we have is what are the risks, those hazards, which Dr. Shelhamer, Dr. Schmidt have already really well laid out. And of course, some of the ways that we could study the, changes to the body and radiation and microgravity is to do what the Air Force did, back in the 1960s when they would take cats. This is still one of my favorite videos ever. If any of you have cats, you always wonder, do cats always land on their feet? And in parabolic flights, the answer is not always, basically. So this is, a short-term version of microgravity. But the big question we had was when Scott Kelly, as was alluded to, went to space for a year. We wanted to measure everything we possibly could because it was a real opportunity to have a genetically matched control on the Earth, who's now also a senator. So we still do annual reviews for some of their blood work, and they're still doing great. Though it's weird to get blood samples from the Senate building in DC, but it still happens just fine. And when Scott was up in space, he actually did a lot of work. He did some science. He did some juggling. This actually looks hard, but it's very easy in microgravity. And for brevity, because this has been work that's now a few years old, we published, essentially an examination of every modality of biology that we could. And I was one of 10 PIs. It was a large collaborative effort, very close coordination also with NASA, of course, and getting fresh blood samples and frozen blood. And we did really an examination of things that move, in concert, which indicate co-regulation or at least coincidence of changes. Do things increase or decrease in flight here in gray? And what are those things? In particular, for example, increases, we could see that actually cognitive measures for Scott got better in flight in some cases, his speed and accuracy. Some metabolites change, microbial species, proteins, cytokines, a lot of things I'll talk about. Gene expression, which you also just heard about. One of the more surprising features is that telomeres got longer in space, which was a bit of the opposite of what we expected. Work with Susan Bailey, but now we've seen that this is actually more and more consistent, and I'll come back to that in a little bit. But this was the first foray of really using omics and genomics, transcriptomics, proteomics for any astronaut really at this scale. But of course, it was one person. So this led to us discussing, well, should we measure all these things again? Should we measure more things and do multi-omic monitoring for astronauts and cancer patients and people in immunotherapy? How do we build context across all these different omics layers was one of the big questions in 2019, 2020. And then, of course, around that corner, then we said, well, we should have an informed consent form and an IRB where then anybody could join. If anyone's going to space, I will make the plug here. If any of you are going to space, please reach out to me. We can enroll you in the study, which looks like this, which is a multi-omics physiological and clinical analysis of spaceflight. And so we built this out as a protocol that built off the twin study to say, well, if anyone's going to space, we should really examine, everything that we possibly can, because we don't have a really good map of what's the most real sensitive feature, or even what's the most sensitive cell type in the body. So this is a list of what we did for what we had a chance, basically, was the Inspiration 4 mission. So I'll jump to that. Here is that they announced in early 2021, and we're heading up into space. So we enrolled the crew into this protocol, jump back to it again, and made it so we could get pre-flight, in-flight, and post-flight collections across a wide range of tube types as well. So it's not just a little bit of whole blood and some basic profiling. We really had a broad battery of tests that we could get from one blood draw, but many tube types to get a really comprehensive analysis of each cells. We could look at apoptosis. We could look at circulating immune cells, epigenetics. Also did some standard clinical lab workups of the blood draw. So this is when we're doing the collection, it looks like a scary amount of tubes, but it's not that much volume, actually. It just looks like a lot. And then also did a lot of collections of swabs, urine, stool, and saliva to also see what changed. And so for the I-4 crew, of course, when they got up into space, they of course took some photos because they're excited because they're in flight. But they also then got to work. This is the Jared Isaacman, the commander, and Haley Arsenault is the medical operations lead, and basically getting ready to start doing some science. The Apple Watch flew for the first time. They did some of Dr. Shelhamer's experiments on there as well for looking at sort of the perception in space, an osteolith. Or basically, measurements of how they're getting spaceflight sickness, which 2 of them did, and we might be able to predict that better in the future. The Apple Watch flew for the first time. Also, the Butterfly device for doing ultrasound imaging flew for the first time. So some very unique firsts about this. Some of this is because the SpaceX medical operations team can work very quickly to say, okay, let's get something flight validated, get it up there, and really within a matter of a few months, we got the hardware approved, got up on working on iPad and up into flight. And so that was novel, definitely on the hardware and certainly on the all-civilian crew. But the thing I want to talk about, especially today, is some of the novelty of different kinds of molecular assays and genetic assays that we were able to deploy for this crew, which we'd never done before. So one of them, and the first one was what's called a multi-ohm single-cell workflow. In this case, we were able to look at each individual cell. So these are PBMCs, peripheral blood mononuclear cells. And we can look at both epigenetics and gene expression. So you can see how is DNA packaged and contained, and how are genes activated or repressed? What happens to the genes, as they're getting into space and when they land back on Earth? And so this is a schematic where you can see each individual cell. It gets dropped into a little oil droplet and then can be put into these single nuclei capture elements that then get DNA and RNA out from each individual cell. and then we can do the analysis once we do the sequencing of these cells. So one of the first questions we had is kind of an older question. It's a question for almost any phenotype or any disease or any sort of state of health is, which cells are changing in response to any stressor of any kind. And in this case, we want to look across each cell population. You can see here from stem cells in your bone marrow and in your blood. Cells, of course, differentiate in your body into T cells, B cells. You can see these different subtypes of cells. And what we found here is actually that the CD16 monocytes had a lot more changes than other cells, and T cells also showed a lot of activation. So we can start to see this burst of essentially differentially expressed genes, or DEGs. What genes are changing the most in different cell types? In some cases, some of these are unique to that cell type, and other ones we see the same genes change across all cells. So if you notice here, there's more gray than there is colors. That means most of the changes are similar across cells, but there are very unique changes in these monocyte cell populations. You can see here, which are part of the myeloid progenitor population. So things that are really regulating part of the immune system and hematopoiesis are changing more so than we've seen for other cell types, which is interesting to see. And actually, one thing that came up, I added this slide just as Dr. Schmidt was talking, is how do we measure all the other parts of the body that are changing? You can separate out individual cells like this. This is one way to look at each cell type. But there is a new assay that we performed actually in this paper, which was cell-free RNA sequencing. So some of you have probably heard of cell-free DNA sequencing, which is a way you can look for early evidence of cancer. One of the companies that does this is called Grail, or there's Freenome, or there's Tempest. There's many companies that do this as a way to do early detection of cancer. But cell-free RNA sequencing is a new assay we've been working with the Development Lab at Cornell to deploy this more routinely. And what's interesting about this is, if you think of what's in your blood, of course you have, you've got all the cells, you think about every time you cut your finger, all the blood, but what's in that blood, and particularly in the plasma, is all of the RNA from all the tissues in your body, but it depends on which tissues have recently, which cells have recently died or released some of their RNA, or which ones are the most active, are all kind of present when you look essentially in the blood. And if you look at the day they returned back to Earth, this is where we saw signatures. You can kind of see it here in purple. What's the signatures for hematopoietic stem cells? We see this burst when they land back on Earth, but it does go back down within a few months later. But this is one of the more surprising features from the study. And actually, to Dr. Shelhamer's point, we don't know enough yet to have this be targetable or medically actionable, but it's interesting because now we can see a version of a whole body molecular scan without having to take samples from all over the body. We can see it reflected in the proportion of cells that are represented in the plasma. Literally, the RNA flowing through their blood is indicative of which cells are stressed or more recently undergone activation. And so this is interesting because this has been discussed, hemolysis or other hematological changes from spaceflight, and we confirm another aspect of this here with the stem cell activation and changes after spaceflight in this crew. So another thing we looked at is, okay, there's also been discussion earlier about brain, brain activity, brain stress on the brain, or the ApoE genes. I really enjoyed that discussion. So one of the things we also looked at was what's called the secretome, or everything that's just secreted into the plasma again. And this is basically what's free-flowing proteins in the blood or in extracellular vesicles. You can see here, these like basically exosomes are called, which are carrying their own protein payload. So we did this before, during, and after the flight, and actually did here, you can see these bursts of different red are from the day that they landed from flight. And these are different abundance of these proteins in the EVPs, or in the exosomes. And we could see that the day they landed was pretty stressful on the body. It's actually returning to gravity, I would argue, is probably harder on the body than getting into microgravity in terms of the need for adaptation. And one of the things we saw was actually alpha-synuclein was a neuronal protein, and it's actually normally found in neurons and brain tissue. So this was kind of confusing because we thought, okay, there's many things that change, but the one that was kind of intriguing was SNCA because it's not normally found outside of brain tissues, and it's brain associated, it's brain enriched. So this led to some questions. We then started chatting with Vivian Mao, who'd recently been flying some space mice, to see, if you could dissect their brains and see the differences. Is there like a blood-brain barrier disruption potentially? But of course, it's really impolite to dissect human brains when the human being is still using the brain. So we couldn't do that. But what we could do is use mice samples, of course. And this was one that had been flown actually on some of the rodent research mission 18. And this is the flight sample on the left and the ground control. And you can see the markers here for PCAM1 actually show this, it indicates blood-brain barrier disruption. You can see more of it in the flight samples. This is representative of several of the other mice brains that were flown. So we can actually see some evidence of disruption of the blood-brain barrier, which, again, is not necessarily bad. It may just be adaptive. It's not necessarily maladaptive. This could actually be very an effective way for the body to deal with the fluid shifts in the body, the radiation. This may just be how the body is reacting. But the mouse data give us some mechanistic insight as to how this is probably happening. And also, one thing of note, the cognition data from Matthias Bosner from UPenn showed that cognitively the crew was still performing their tasks fine. So we think this is more an adaptive response and not necessarily anything pathological. So I'll close with some other data from the crew that we also did the largest so far to date astronaut microbiome study. So 758 samples, 10 parts of the body done both DNA and RNA and in replicates and before, during, and after the mission. And one of my favorite figures from the papers that just came out today is this one, which shows each of the crew members and then each of their sort of body parts and where we see microbes moving. The thickness of the line shows how many microbes moved either within a crew member or even between crew members. And you can see that, there's microbes moving between all of us. Even if you go into a crowded elevator, you'll get a little bit of this. But you can also see some people, some of the crew members became more like the capsule, like crew member 001 got a few bit more of the microbes literally from the wall of the Dragon capsule, which was kind of intriguing to see. But there is this continual movement of microbes within and between the crew members, which has always been occurring, but now we can actually quantify and map it out much better really than we ever have before. And so this is another sort of paper that just came out today that is an interesting map of how microbes move and the difference between the DNA and the RNA, because we did both here. We can see which microbes are actually active in spaceflight, which we see some pretty clear differences. So this is a quick summary of some of the I4 data. It's worth noting that a lot of the same discussion were just sent off to Congress, as also to NASA for the decadal survey, which I was one of the co-authors on, is that we need to perform more of these in-depth and multi-omic and longitudinal studies to learn about what changes. And one of the things I'll close with, I'll come back to at the end, but just today there's about several hundred research teams and actually 24 of the papers use data from the Inspiration 4 mission to help build out this new package of papers that came out literally today, literally like 10 minutes ago or 20 minutes ago, it finally got online, is now online in a really big data portal. So one of the things that we looked at earlier, and we can now look at it more at scale, is really this question that Dr. Shelhamer just brought up, is differences between crew members are really pronounced even for these cytokines. So this is a lot of dense data, but you'll notice that the blue line is Scott Kelly from his year-long mission. But if you look at other 6-month missions, these are box plots that show the range of human variability, whether it's pre-flight or in-flight days, FD, or after returning back to Earth. And you can see in some cases, Scott Kelly, in that data, he's very flat. He's or the most flat really of anyone, whereas other crew members have dramatic changes when they're in flight. So what this shows, we've published now in multiple data sets, is just like every precision medicine on Earth, you have to apply the same principles to what you see in flight, is that there are these really significant differences between sexes and also between crew members that we're starting to tease out for the first time. So again, we've only had about 695 people that have ever been to space. We have data now on maybe about 64 of them in this SOMA atlas and the SOMA portal. And we don't have the rest of the data because like maybe about 25% of them, 30% are Russian, which we're not going to probably get anytime soon. And some of the data is just now have been lost to time. But what we've built out in the past few years is something that has been inspired by the UK Biobank. So this is, I think as most of you probably know, a really extraordinary effort to link genetic data and molecular data to the phenotypic data and the medical records. So you can now look at a question of what is an association for, say, Alzheimer's or hypertension. We can start to link the genotype and the phenotype and the molecular and the physiological data. And so this is something we built upon now as an aerospace medicine biobank at Cornell, which just again also launched today. And we wanted to make it so some of the same questions we had when we first did earlier studies is, it'd be nice to look at, I see a gene or I see a protein change. Well, how often does that change? And so actually as a quick preview for today, there's this bank, and then I'll show you kind of the portal. So there's a portal here, which kind of shows the atlas. You can still see my screen, right? I think here, just to make sure. And then if you want to, you can type in any gene that you want and see, for example, how does it look at across, the multiome data across different cell populations. You can look at skin data, the cell-free RNA data I just mentioned to you, the cell-free RNA data from the Japanese Space Agency and some of their missions, cytokines, exosome data is all listed here on the left. And you can also, one of my, kind of favorite things you can also do if you want to look at, say, differential expression comparisons between, say, multi-genes at the same time, or sets of genes. Here's all the data we have from human spaceflight studies showed as dot plots, but also we've uploaded all the data from GeneLab. There's great work by Jen Maiden in our lab. It's all the mouse spaceflight data. So liver, the heart, dissected tissues from other data from GeneLab from any mouse that links to the human ortholog. So you can look at both mouse and human data at the same time to really just answer this question. If I see something weird in my spaceflight sample or any sample, how often do we see it? And is it significant, and how often has it been seen before? So this gives us a way to start to do that on this data portal, which I'll put a link in the chat, but it's now online and linked to from here. And the other thing that's exciting about this is we now have consented spaceflight samples that you can begin to request if you want an aliquot of, say, urine or plasma. The crew's been consented for this, and we're launching this out over the next month or two, so anyone could request a sample. But actually, of note, half the samples will be being for long-term viable frozen cells, because I'm sure there'll be technology in 10 or 20 years from now that's better than what we do today that will wish we had long-term bank samples, which we will have. The other thing I'll just note is the Olivus is another program like the UK Biobank in the United States. My genome is up there, and you can do things like look at my DNA results, look at my measurements. No database is perfect, of course, because, for example, it thinks that I really would not like cilantro, but I actually love cilantro. So I think, we know that the database itself is not what's important. It's about building it, getting it to be longitudinal, and getting people to have access, and getting the models to be better. Because right now, according to the Elvis program, I don't like cilantro. It's literally probably one of my favorite things in the whole universe is cilantro. And I know some people it tastes like soap, and I feel bad for them, but I love cilantro anyway. So, but what's exciting is these models can and will get better, and the same thing is true for our models of spaceflight and precision medicine in spaceflight. So another thing we worked a lot with GeneLab is the OSDR is the Open Science Data Repository for NASA. So a lot of these data from this mission are now all online. And as I just showed you quickly, this is a screenshot in case the internet was down, but there's data port. You can kind of click here and see if you want to play with any of these data for any gene or protein. These are all now online. So closing in the last sort of few minutes is, what's next? Some exciting things that are coming soon is we did try zero gravity sequencing back in 2015 for the first time. We published this in 2016 and showed that it was definitely possible. And then Kate Rubins managed to get up into space and actually show that indeed you could do it with a nanopore sequencer in 2016. So we've been building on that and actually getting some of the later hardware, which is now called the Mark 1C, which is a device that looks like this, to also get this flight validated. So you could do blood draw, library prep, and sequencing to measure cell-free RNA, telomere length. You can measure really infections if they might be present. So getting this just got hardware flight approved, and hopefully this will be flying on one of the upcoming missions very soon. And the other thing that you can get from the single molecule sequencing data, this is Dr. Overby, who's a postdoc in the lab, who's now starting her own lab actually, is you get the signal information as DNA moves through a pore. You can see what base is present, but sometimes you get these modified squiggles. And so we want to use this data to look at modified bases as well as native nucleic acids. And we're basically planning to do this for some of the upcoming Polaris missions, and eventually this will go on the Starship. And so to keep it to about 20 minutes, I'll close in about maybe 2 minutes. So I want to just on philosophical endings, think about going from measuring biology to defending biology. And some exciting work in the lab is thinking about ways to get armor inside of the cell. So some work led by Jen Kim in our lab is actually do epigenome reprogramming for radioprotection. So we try to build a shield inside human cells so they could actually activate DNA repair genes in advance of a radiological event. In this case, actually, this is the zero-G control. As you can see, some of the cells died after we thawed them, but some of the cells that were irradiated with 10G, of course, none of them lived, but then some of them with our basically CRISPR-Cas9 system, we recovered them, went from lethal to actually at least some of them surviving, which I think is fascinating and kind of links to some other ideas that have happened in the literature where, for example, using CRISPR systems to cure sickle cell disease by reactivating a gene that's present when you're active as a fetus, the fetal hemoglobin instead of adult hemoglobin. So I think it's a fascinating era in which we live that we could start to think about controlling gene regulation and tweaking it instead of doing just drugs. We could also think about gene therapy themselves as more transient drugs. And again, this is borne out a lot by the literature. And of course, we also have some fun work we do with tardigrades. We're trying to get these tardigrade genes into human cells, which we've shown here. And actually, they do also help with radioprotection. It's a bit more, hard to do, but you can actually use them as well. And then finally, some of the other engineering is I don't want to just think about human cells. We're also looking at how you could get microbial and plant even systems that would work on the moon. So we'll be getting some lunar soil in next regolith in about a year or so. And we've been trying to take lessons from really all creatures anywhere. So this includes creatures at the bottom of the ocean, some of which we found these cyanobacteria that can eat carbon dioxide so far the fastest of any bacteria we found. And then also work a project called the Extreme Microbiome Project, finding creatures, just odd creatures in different places around the world and taking evolutionary lessons from them to help us prepare for trips to, say, Mars or Venus or even just living in space. And so we've published some of this work that shows polyextremophiles really give us interesting lessons. And this work is ongoing across many other sites from different places around the world of different pH. pressure, temperature, and radiation levels. And I think we'll take lessons from all those creatures whenever we can, basically. So I want to close, of course, and thanks to this. None of this work would be possible without having a phenomenal and wonderful team in the lab that makes it the only way possible for me to be here at all. And of course, funding and support from logistics, especially from NASA, but also logistics for SpaceX, some support from Trish, World Quant, Pershing Foundation, and the NIH. Finally, gratitude and thanks to many people and teams, including people who I've collaborated with on this call, including Michael Schmidt. And finally, I want to close with one last thought of a paper that just came out today is this idea of maybe doing a 2033 mission that we could go around Mars and Venus and back in 18 months. And I'm currently fundraising for this mission if anyone would like to come help or just chat about the science about it. But what would be fun is you could drop off probes at Mars and Venus and still get back in 18 months, and it'd be kind of fun. So the last thing I'll note is these did all just come out today, and there's the SpaceX, this paper just came out also with Dr. Shelhamer and team here. And also, so this is now about the Inspiration 4 crew, this data portal, and there is also this landing page, which is now also online. So I'll put this in the chat if anyone would like to see it, but we're very excited. These have just come out as of today, and we'll keep a little on time and happy to take any questions.
Excellent. Thank you, Chris. Just as with Michael's talk, I am full of questions. I'm gonna drop two of them and not ask for your comments, but just to make the point that there are, but there is one that I will ask you to comment on. One of them is that you were talking about in going from measuring to defending and making deliberate epigenomic changes to hopefully enhance the ability to respond to the stressors of spaceflight. Needless to say, that raises a huge number of ethical questions. I don't want to really get into that today because we might do a symposium on spaceflight ethics, and there is a whole subcategory of medical ethics having to do with the ethics of spaceflight, human spaceflight. And the other thing you mentioned was you showed a slide about distribution of microbes within the Inspiration 4 crew. And I know you've done this with other situations, other settings, including urban settings as well. That raises the question, which I will not ask you to address at the moment. You can do it later if you want, as to whether or not we should take deliberate efforts to provide a microbial variety in long duration space flights, because it's my understanding that as space flights become longer, the microbiome, the biomes of the microorganisms of the different crew members start to become more and more similar to each other. And whenever there's a reduction in biological variability, that's not necessarily a good thing. So you might wonder whether you want to take up jars of dirt, microorganisms that you open at periodic intervals during a mission to keep the microbiome fresh. Hold those thoughts.
Great questions.
Okay. The thing that I want you to talk about a little bit more is was getting at the concern that I have, that I also addressed to Michael Schmidt when he was done talking. The idea that this is so complicated, the different networks, the different interactions of these pathways, metabolic pathways, gene expression pathways. In how many cases can we really claim to understand something to the extent that it would be actionable? But you started, Chris, to get at something that was more along the lines of whether or not there's a more general indicator of overall healthiness, adaptability, well-being. Is it inherent? Is there something that we can glean from the distribution of things that we measure in various body fluids, for example, where we could say, well, gee whiz, I can't claim to understand that in detail, but I kind of like the pattern of it. Or conversely, I kind of don't like the pattern of it. Something is telling me that this is losing resilience, losing adaptability, losing its ability to respond to changes in environmental conditions. Anyway, comment on that as you wish.
So many great questions. I think the first thing that there's a lot of questions about the ethics of spaceflight, which I put a link in the chat. I wrote, I've been thinking about this for a long time. I wrote a whole book on it, so it's there if people want to read it. I think it would be a great point for a whole symposium of what are different kinds of therapies, pharmaceutical, genetic modifications. And I think when I started talking about this like 10 years ago, everyone thought I was a crackpot. I still am kind of a crackpot, but now it's one that has a lot more evidence behind it in the sense that there's thousands of clinical trials using subtle variations of gene therapies or epigenetic modification systems. So it's a really exciting time because what used to be science fiction is now really, in the clinic and curing disease. So I think it opens up really interesting big questions, but how and when you use it have to go through, normal safety profiles for like any therapy has to go through various phases of clinical trials and safety and efficacy that before you would ever do it, especially for anyone leaving space where they'd have fewer resources into space. And then for the baselines, and just to at least leave some more time for other speakers and talk discussion, but what we all take for granted is when you go to the doctor, you get blood work and you have a normal range, and you just know what that in theory should be. And we're just sketching that out for the first time for astronauts. A lot of it seems to be, I think, adaptive, not maladaptive, and it's the body just coming back. But as I showed in that one figure of the box plots and violin plots, it's a wide variability. And I think we're just teasing that out now. So what's good is we're finally getting in this state of having dozens of data sets, almost 100 soon. That'll help us really figure that out and then get customized countermeasures, customized monitoring as well.
Okay, so it sounds like you're optimistic about the ability to not only refine the kinds of work that you guys are doing and find more actionable pathways, for example, like the kinds that Michael Schmidt was talking about, but also to get some more kind of broad-based indicators of whether things are just going off the rails.
Yep.
More generally. Okay.
Yeah. And hoping, and something like, for example, we know is a good example, like when Scott Kelly had the really bad rash when he got back after a year in space. He has levels of IL-1RA were the highest we've ever seen for any astronaut out of over 60. So that's probably what was at least related to it, but does it cause it? We can't really say yet, but at least we start to get these clues of, well, he had a really severe reaction and the highest level of any cytokine we've seen that's pro-inflammatory, so it's probably related. But do we target it is a bigger question that's like probably not yet. So, but at least we know what to start to look at and keep an eye on.
Right. Yeah. Cause and effect could really get you into a bind here. Okay. Very good. Okay. Thank you, Chris. Now, Christopher Bradburne has been very patient and hopefully has been biting his tongue, jumping in here, wanting to hammer you guys with things about things that you've said. We'll give him a chance. Go ahead, Chris.
Well, I don't know about hammer. This is all incredibly interesting and really excellent presentations, well put together, and it's really neat to see the impactful research at the bleeding edge of human spaceflight that you just presented. I've been in the omics field for a while. Most of my work has been for DoD and DoD area sponsors, and so familiar with the challenges of precision medicine, familiar with the challenges of N of 1 studies that Mike Schmidt talked about, as well as Chris Mason with the twin study, and also very familiar with the challenges of all the different omics and measuring them and how the technology changes over time. So Chris Mason mentioned the nanopore sequencer. Love that technology, but I think those of us who use it know that it does change over time. And understanding that some of those changes and understanding what's possible is one of the important things that we want to try to I love to use the word future-proof. How can we future-proof future studies? Chris, really appreciated you mentioning the consented spaceflight samples that y'all will have and the SOMA to the SOMA interface for just such a reality of being able to go back and look at things with newer technologies as things change. One of the things that that does change over time is also how we confer risk to human disease and how we do something called genome-wide association studies. So a lot of the genomic medicine data out there linked to genetic markers for human beings has been done over 25, 30 years, and initially was focused on the single nucleotide polymorphisms that Mike talked about. But again, over time, technology changes. And one thing that we do know is that SNPs don't confer, they don't account for all the risk, right? for chronic diseases for anyone. I think macular degeneration is the best tie to any SNP variant for risk prediction. So it really is that environment gene interaction, and that gets to a point that fancy word geneticists like to use, epistasis. So you've got a lot of things going on. You've got not just one variant, especially for the complex and chronic conditions and chronic or complex traits, but you've got potentially tens to hundreds of genetic locations that are interacting with each other and with whatever the environmental stimulus and environmental factors are. How do we get to an understanding, a real true mechanistic understanding when you've got all that noise, which isn't, to repeat something Chris said a second ago, it's not just about, it's not just the single variants that are providing you that risk prediction. So is there actually, how do we actually go from our current understanding with as the technologies change to a better understanding? And the digital twin concept is really, really interesting. We're doing some of that work here. Digital twins for human systems. I would also say there's efforts coming out of the toxicology community for adverse outcome pathways, which are trying to link stimuli to systemic effects for chemical toxins, but actually there are folks in my section who are also trying to apply that to infectious disease. And what that tries to give you is some way to explain what's going on. But key to all this is going to be the data. So all of us in the UK, biobank data is going to be really, really important moving forward, and feed, I think, the digital twin, sort of the digital twin concept for real N of 1 studies. So, the issue with spaceflight, and Chris said it, 690 something people who've flown in space. That might power a GWAS, a genome-wide association study, but it's really tough because you've got low numbers of people. So really excited to see how things are going to move forward, combining all the things that y'all are doing and combining all the sort of the ground level foundational research that not just our nation, but other nations are supporting. So that gets me to a question. And I guess I will start, since we're talking about genomics in space. The question I'll pose is actually to Chris Mason. So genomics are expensive to do, and they're also complex to do, and they take a lot of time and effort. And basically, it's a snapshot, right? It's a single snapshot in time, whereas your physiological response or your physiological signaling might be fluctuating depending on what the signal is. So in terms of that, like one of the things that to me is really compelling, especially if y'all are flying like the Apple Watch for the Inspiration missions, could that be something you use to, like, when do you actually trigger a genomics measurement, as opposed to just pre-launch, in flight, and then post-landing? Could you use like the Apple Watch to indicate a period of stress and then trigger a measurement? What do you think is a good cadence? And how do you optimize the use and the measurement opportunities you have with omics?
So before he answers that question, I love that question, because clearly one of the things that we're finding in macroscopic physiology, heart rate variability, those kinds of things, is that variability in an individual over time is actually a strong indicator of health and well-being and fitness. And if you were just to take a measure, for example, of your heart rate once a week, that would tell you something pretty interesting. But if you were able to get your heart rate continuously for 8 hours, that tells you tremendous amount more, the ability. So yeah, it's a really good point. To what extent do we understand the normal variability? I mean, gene expression is changing every time I take a drink out of my coffee. I understand there's a gene expression changes responding to that. So how is that? How can that be accommodated or recognized? Dr. Mason.
And there's a great question. So Mike Snyder has done a lot in this space. I've probably heard his integrative personal omics profiling, or IPOP, which sounds like a popsicle. I always feel like I want a popsicle every time he says it. But the idea is, if you just monitor people over time, you learn a lot. And in particular, he has a really great slide he almost shows in almost every one of his talks of, we think of 98.6, or what is a normal temperature. And there is no like, there's a median, but there is a pretty wide range. Of some people's normal baseline is 97, some is 99, 99.5. Some people run hot, if you will. And so you have to know what your baseline is to know if you have a fever. And so I think these measurements, I always err on the side of as many as possible, whenever possible, at least in the early stages, for either for a new drug, for example, we do this, or for a new treatment. In the case of spaceflight, it's basically a fairly unknown stressor. So we measure everything. I like to say, when you don't know where to look everywhere that you can, because you don't know where is going to be the biggest change. And then eventually, hopefully in 10 years, we won't have to measure everything all the time for everybody, much like what we do with blood work. We know there's key signaling molecules and glucose tolerance tests. And, there's a few tests we can do that tell you a lot of information about risk of a disease, or how a drug will work. And so we're not there for human spaceflight yet, but I bet if we start getting starships with 20, 30 people or 100 people at a time, and we start really getting into the thousands of astronauts, we'll have a pretty good sense, I think, relatively soon.
Will the technology ever permit something akin to a continuous measurement, continuous assessment of gene expression changes?
I think so. So the same, like you could do like a watch where you have the heart monitoring, or pulse ox, or V and VO2. The closest thing is continuous glucose monitors do this for glucose, but you couldn't, in theory, have samples come out and actually could be continually characterized. There is no automated way to do this. There is a device by Oxford Nanopore called the Voltrox, which was trying to do this. And so I think technologically it's not impossible. It's just right now there's nothing on the market that will do it for you. You'd have to do a blood draw, prep it, and sequence it. But we're going to try that on one of the next SpaceX missions, and see if we can, at least as a proof of principle, show it's possible. And also, there's a lot of information in blood, I think, is the reason we take it. You can look at risk. Is there an active infection? Sometimes it's not because you've seen this specific virus, but because you have the signature of the immune system that has seen the virus or the bacteria that tells you likely what it is. And Charles Chu and others, we've published some of these papers as well. So I think, every molecule tells a story, and you have to listen to it as much as you can.
Yeah, that's really good, because the idea is we talk about actionable intelligence, and especially if you talk to, the spaceflight providers, especially in the NASA context. Science is good and research is good, but really, why are we measuring it if we don't know what to do with the information? And in this case, how often do you have to measure it to be able to do something actionable in the course of the mission, which would be extremely powerful to be able to do that.
Yeah, exactly.
Yeah.
That would be, which I think is all.
Especially if you had early warning indicators, something is going on, the body is responding to something, and we don't yet see the overt manifestations of it, but the body's doing something to respond to some kind of process. Michael Schmidt, I see that you're prepared to speak.
Well,
I was going to turn it over to you.
So, these are really great questions, and one of the things I wanted to highlight on here is that because we have such limited measurements, that to Chris's point, collecting as much as we can on these missions is as frequently as we can is going to be really critical to develop the kind of data that's going to point us toward those kinds of measures that may be significant and require a countermeasure, and those that may not. But I would also say that what we're also looking for is, let's say, sentinel measures or surrogate measures, right? So there may be patterns that are emerging that where in the future we won't have to measure everything, because there's sort of a covariation that takes place between many measures. So there might be where we can simplify this and get down to a surrogate measure, or a set of surrogate measures. I think the other thing that's important is that there's the ease of measurement is going to become really important. And one of the things that would really help us in space would be a finger stick approach to, let's say, the transcriptome that's reliable, so that we can measure that frequently enough. That's the thing that's always changing in response to the environment and in response to nutrition. response to the G loads, radiation exposure, etc. That would be highly informative. But also, I wanted to add, Mark, you and I have talked about this quite a bit. It's really important for us to marry all of the molecular data to physiologic data and to the phenotype. And so that's the next advancement in the field that has to be really central, where groups like all of ours are collaborating to try and advance that cohesiveness between, what are the physiologic measures? So even heart rate variability. Are there particular molecular measures that are the antecedents toward that declining heart rate variability? And we need to be looking for markers like that. But I would say that's going to require a lot of measurement over some years here, so that we have enough data to do that. Then I'll just add one last thing about to Chris Bradburne about the digital twin, which there's probably 2 points I'd like to suggest. I think one of the key things is because these can be unstable twins if you try to model the entirety of it. And so this subdomain approach is really useful, where you might model a component like the aircraft engine isn't the whole airplane. And so for humans, we might model the brain, we might model muscle, we might model bone, heart as separate twins that are much more stable. And we would try to incorporate those measures with the largest effect sizes. Let's say the fewest inputs with the largest effect size would be an ideal kind of a model, or the kind of a twin that one could build. But again, all that's going to take extensive amounts of data in the space environment to be able to build those properly. But I think they all start with Earth-bound data sets, right? Those extensive data sets that are built from all of the work that's come before on Earth. So that's a few strings of ideas, but in response to what some of you have been saying, which have all been fantastic points.
Yeah, I'm glad you mentioned again the genotype phenotype idea, which I've been expressing as actionable intelligence, right? Like, what does this tell us about the body and the response and the physiology? But back to,
if I can just add one last point,
yeah.
Mark. Yes, the phenotype, I think, is the crucial thing to all of this, because we don't just want to measure markers and then move markers around with our countermeasures. We want to impact the phenotype. And I would say there's several different types of phenotypes, right? There's a disease phenotype, but there's also a performance phenotype. So we'd probably be looking at a different phenotype if somebody's going to have to be capable to be in a remote setting without resupply for a period of time, or if they're going to need to do an extensive EVA. That's more of a performance phenotype. So how does that particular load impact the individual's capability and our need to develop countermeasures tailored toward that operating environment?
Yeah, let me do you one better. Is there an adaptation phenotype or resilience phenotype, which not only tells you about performance, but tells you about the ability to alter that performance or maintain that performance under stressful conditions? And
Yeah.
I'm guessing the answer at the moment is no. It's not identified, but it certainly would be useful to have that.
Mm-hmm. Yeah.
Because I think about a Mars mission a lot, and a defining characteristic is that something is going to happen on that mission that you have not thought of. So it would be good to be able to select for adaptability, which of course space agencies do now anyway, implicitly. There are other ways to assess that, but not yet in terms of a genotype. But back to Chris Bradburne's.
And the microbes too. You brought up the microbes too. Is another way I think
Yeah.
the ability to also add that in. You brought that up earlier, is keeping the diversity. So like the human and the microbial adaptability is a good point you brought up earlier on that as well.
Yeah, actually that's an intro I never thought about. Right, the ability of the microbiomes to change would confer the ability of the humans to change, because the microbiomes help implement a lot of the metabolic physiological processes. Okay, good point. So that could be a connecting aspect.
Can I add one point
Sure.
to what Chris? I want to tie one thing together, if I might, and if I dare show this slide again. I want to tie something Chris said together with with what I had said earlier, because and Mark, you had kind of raised it. So okay, so if we go back to this, so Chris had mentioned something earlier in some of the data that we have from Inspiration 4, etc. So remember when I talked about tryptophan, it's an amino acid we ingest every day. This is how we make our serotonin and melatonin. It affects mood. In fact, I mentioned these enzymes that make this highly inflammatory, and especially with regard to the central nervous system, quinolinic acid. So in the data that Chris showed from Inspiration 4, and then the converging data from some of the other missions, I'm talking about interferon gamma for just one moment. So Chris, you showed a very significant increase in interferon gamma, but a very high individual variability.
Yep.
So, we would look at this, and this goes back to your question, Mark, where do you act, and, what are the countermeasures, and what do the genes tell us? Well, this comes from some gene expression and protein data that's basically saying, well, the interferon gamma went up, so this enzyme is activated, and so that dietary tryptophan is pulled into this quite neurotoxic, it's one of the excitatory neurotoxins in the central nervous system. So we would look at this and say, well, there's immune variation in space, and sometimes it's adaptive in a positive way, getting back to our earlier conversation. So we may not want to blunt the effect of interferon gamma because we really don't know what that effect is going to be in space. So this is where we would say, well, let's not inhibit interferon gamma because we just don't know. But we do know the effects of reduced conversion of tryptophan down to serotonin, because you get sickness behavior, you get withdrawal from social interactions, there's a range of mood phenomena, depression that can go with that. And then melatonin obviously associated with sleep, but also mitochondrial function, etc. So that's where we may say, okay, we understand interferon gamma. It's a little uncertain right now to deal with it, but let's not load the system with tryptophan here. Let's give the 5-hydroxytryptophan. So at least we'll fuel the serotonin pathway and the melatonin pathway and allow them to naturally upregulate. If we just say we're going to give more serotonin or more tryptophan, we may fuel this pathway further because this interferon gamma is active. So I just wanted to kind of contextualize that in relation to what came out in the untargeted analysis of the immune proteome and the transcriptome, because this at least says there's a phenomenon. Now we can find what we think is an appropriate workaround, and this is usually quite a safe workaround and a fairly well-defined workaround. So I'll just stop there, and we can move on if you want, or discuss that further if you like.
Yeah, let's hold that because we want to get to some questions from the audience, and Malika is all queued up and ready. But I did want to turn it back to Chris Bradburne for two things. First of all, I wanted to wrap up the comment that led to this discussion. I think the basis of it was something that Chris, you and I discussed a number of years ago, which is that one way to potentially address the sampling, essentially the sampling rate issue, or how often do you get blood samples, saliva samples, whatever they might be, and how do you make sure that you're measuring them under comparable conditions so that changes you see are not just due to the fact that somebody's tired or just ate or just exercised or whatever it might be, but they're really changes due to space flight per se, the other aspects of it. And of course, one way would be to try to standardize those conditions. So you might do that artificially by setting off an alarm, setting off something that they have to the body has to respond to, and then it may be artificial, but at least you've kind of made something that's constant, that every time that happens you get a blood draw, whatever. Obviously some logistical issues for that, but it's the same idea they have with a fasting blood draw, right? You try to normalize the conditions. I'm not going to ask for a comment on that, but Chris Bradburne, I want to see if you had any other pressing issues that you wanted to get into the conversation before we go over to Malika.
Sure. In the interest of time, I'll make it short. Just maybe a follow-up question on adaptation point that we were talking about earlier. in the military, we talk a lot of, well, we used to talk a lot about a term we coined, the gritome. So it kind of, how do you actually measure that desire in the human performance perspective, that desire, that grit of somebody has, that when they're disadvantaged in some way, that they actually overcompensate in a motivational way to actually accomplish the mission.
I
Some
love
of those
the
things could, yeah,
I love that
No.
term. Great.
the gritome. How do you measure that grit? So for example, in the serotonin example, you might have all this physiological data that indicates, oh, this person might be at risk, but actually that could be motivating them to overcome that deficiency. And that's sort of above and beyond just the biochemical and physiological data we might be collecting. So how do you incorporate that in your measurements? And we all know astronauts, you know, a lot of them are going to probably have elements of that gritome.
Yeah, and remembering gene-environment interaction, to the extent that there is a gridome, it's going to be, people rise to the occasion. That's another way of putting it. But so it's some combination of the environment and something that's inherent.
Well, and how does the gridome or the grindome even contribute to some of the deficits, right? So there may be some molecular patterns that provide the capability to act on that, but there may be some findings. So we did some molecular profiling, the NFL Combine, before the NFL draft, and these were all top draft choices. They were all at top universities. So it was some of the top players in collegiate football. And when I presented our findings of their molecular profiles at the Metabolomics Society in Dublin a few years back, one of the guys who runs a laboratory in Germany said, those lab profiles looked worse than some of my sickest patients that we see in the laboratory. And he said, how do you account for that? I said, yeah, so I think part of this is the heavy loads of training, right? And the heavy loads of execution in extreme environments. And so this is just to add to your thought, Christopher, is, I think there's that considerable behavior and capability that's been developed. And there's intrinsic motivation and force of will, et cetera. And then there's underneath that, these molecular elements that could support that or become dysregulated as a consequence of that. And so I think if we don't measure that behavioral phenotype in all of its permutations, then our molecular, and this gets back to the concept of phenotype, right? Then our molecular doesn't have the context it needs to interpret it and also make decisions derived from it. So the clinical decision support is lacking enough context.
The psychology becomes very important. Yep.
Yeah. No, I like that idea. You can become your own worst enemy.
Yeah.
And, NASA, for example, for professional astronauts, they want people who understand risk, who can make the risk calculations, have exposed themselves to risky conditions, but they don't want necessarily daredevils. They don't want people who are reckless, but they want people who are not afraid of risky situations. So that could be, would that be, certainly it's reflected in a phenotype, if not a genotype, but we're going to have to stop at that point for that discussion. Thanks to the three of you, but you're not done yet. Malika is fielding us some questions from the audience.
Great.
Yes. Thank you all so much. What an interesting and fascinating conversation. As a behavioralist myself, I am really excited that we are connecting the molecular changes that we're seeing to phenotypes and behavior. And so along the lines of a small n population that we work with in spaceflight, we have a couple questions from the audience about analogs. In the spaceflight community, we talk about analogs a lot. Because we have such a small n, it's important to get data from other locations. So a couple of members of the audience have asked about the study of extremes here on Earth, terrestrial populations that we can use, whether it is elite athletes in extreme environments, like Dr. Schmidt you just talked about, or polar or cave microbiomes, or any of the NASA specific analogs, whether it's high seas or Hera, and what information that can provide for omics.
Yeah, I guess I'll start. I think so. We've been doing Yeah, and actually, Dr. Schmidt does this. You have a whole company that does this as well in terms of like the extreme medicine. Just quick comment on one of the papers that just came out today. We compared some of the non-coding RNAs that regulate telomeres and probably make them longer and protect the DNA damage. We compared it to climbers from Mount Everest, and we see some of the same signatures there, and that's also stress and more radiation that we see. So I think there's a great case to be made for the analog astronaut community. The expand program that Trish is running, it does some of this. There's a lot of groups trying to just get data of other, extreme sports. We're actually doing, I'm running the New York City Marathon, and some other people in my lab are in this year in November. So we're definitely going to get blood and urine and saliva before, but then we're debating, should we take samples during the marathon, which probably seems unwise because people will be bleeding all over the place. But anyway, there's great discussions in our lab about, like, for the same point, like we need more context. And those are great examples of places we can get it. So I think we'll definitely do pre and post the marathon, but maybe not no blood during. But saliva is easy. You could do that. Anyway, I'm sure Michael have some comments.
Yeah, I think so the hard one is radiation. Some of the radiation, let's call them analogs, have been looking at commercial airline pilots, military pilots, and astronauts. And there's been some work where, because that's a surrogate for different levels of radiation, looking at ocular changes, and they do seem to scale with, location and duration of exposure. But we don't have good radiation analogs on Earth. We don't have microgravity analogs on Earth. We have buoyancy, but not microgravity. I've always wondered if we resolve the microgravity problem in space, how many of the problems that we see would just simply go away? And that's an unknown at the moment, but it's one that I think all of us are asking continuously. But I would say one of the things about analog settings, and so in work we've done with Navy SEALs and Delta Force and other really high performer environments, there's always a question: how much analysis do you do to try and get as much data as you can? Let's say heat tolerance data, or when do we test the individualized interventions in those extreme environments? And so let's say you have 12 SEAL operators, and once we start an intervention, whether it's a generalized intervention or an individualized intervention, now we've changed our ability to gather all the data about that operating environment. And so we're always wrestling with the ethical part. When do we just study it? And when do we actually study the doing something about it on an individualized basis? So, for example, Stan and I think Dingus was part of that. They did a 14-month expedition to Antarctica, and they did MRIs before, MRIs after, but they were looking at brain-derived neurotrophic factor, which is one of the things we've looked at for a lot of years because we have concerns about that in space. And they found over the 14 months that BDNF levels went down substantially, and it was associated with changes in hippocampal volume and some cognitive measures in those individuals. So that kind of study in an analog is really crucial for us to have. But how many of those do we do now before we start thinking ethically, like, well, we have to now start doing something to intervene and try to help them from that deterioration or dysregulation taking place. But then once we do the intervention, now we've changed the whole observational part of the research. So these are just musings that we always face. But the analogs are, I think, crucial, but they're limited, right? Especially on the gravity and radiation side.
Yeah, you're right. If you want your Nobel Prize, figure out how to simulate 0 G on Earth.
Yes.
There are so many manifestations of it, including the fluid shift and probably direct effects on cellular function, including intercellular signaling that we're just beginning to understand. And there are some, as you say, there are some analogs, but there's nothing quite like chronic altered gravity.
Yeah.
So you don't have to comment on that. Malika, next question, please.
So, Dr. Mason, you mentioned that we have this really amazing Nature package that had just came out today at 11 a.m. I think that it was added into the chat, so I hope that everyone gets a chance to check it out. But I know we're in our last couple of minutes, and so I want to end on a happy note, and I want to talk about what does it mean to do teen science? This project required the coordination of so many different research teams, different PIs, different governments, institutions, commercial and government agencies. And I think that many of you on the call have been deeply involved. And just to talk a little bit more about what does space and working in teams mean?
Sure. I think, thanks for the question. I think it is, yeah, like some of the biggest questions you can only answer as a team. The old adage that if you want to go fast, you go alone. If you want to go far, you go together. And I don't know where it originally came from, but I like to combine them. It'd be great if we can all go fast and far but I do it together and all, in concert. So I think there is, I think a lot more, just a bit definition, some things like, for example, if you want to compare between multiple crews across the world, you have to coordinate. If you want to, since the data are so precious and rare, we have to be able to compare between ESA and JAXA and NASA and now more recently the commercial crews, SpaceX and Axiom and Sierra are starting to get their crews. So I think team science at this point, actually, it used to be team science between academic institutions and then maybe between countries, but now it's public-private partnerships. It's really have everyone with the best interest of exploration in mind and sharing their data. And it's always the thing that takes the longest is the lawyers, of course, but someone says, well, who owns the data? Can you commercialize it? So it took a good year to set up an agreement between Cornell and SpaceX. But then now it's done. But, so I think we're also going to release a lot of our documents and IRB protocols and make it so people who want to join in can start to not have to wait a year of legal negotiations, but just start to help. So it's sometimes it seems like legal is the hardest thing. It's not the science, not the scientists. Most people want to share and collaborate. It's really just making sure that if someone wants to make a product out of it, commercialize it, that gets much more complicated. But in general, team science I think it's been really great. I've been very blessed to work with a lot of amazing people and want to keep doing so for as long as possible.
So the team, sorry, the legal, ethical part and who owns the data, who owns the samples, is not, and I'm going to turn this over to Michael Schmidt in a minute because I know you have some things to say about this because Michael and I are both heavily involved in the development of a human research program for commercial spaceflight. And among other things that you would have to deal with is if you are a paying customer on a commercial spaceflight and you're doing a research project that is sponsored by a drug company or NIH or both, and you've got some data that might actually lead to a product or tell you something about how people are responding to the spaceflight experience, a lot of people want to get their hands on that data and possibly keep other people from getting their hands on that data, including you, the individual from whom the data was extracted, the people funding, the organization funding your research, the flight providers, whoever else, the university, if you're a university researcher, the list goes on and on. And so it is not at all trivial to figure that out. What we're hoping to do in this field is start out on a good foot and set a good precedent by saying to the extent possible, these data should be widely shared so that the community can mine the data because we don't even know the right questions to ask in many cases about the human response to space. But that's a little bit unrealistic because there's a lot of proprietary information concerned. Dr. Schmidt, would you like to say anything more?
I'll just add a couple of thoughts, and I just really appreciate all the interaction and feedback. This has been just fascinating today. And one of the things that I think is really important about this, and you mentioned, Mark, the Human Research Program for Civilian Spaceflight, and that we're working on together. And one of the things that is a goal of that is to try and harmonize the methods to the greatest extent possible through a kind of a set of comprehensive measures that use similar methodologies, right? Not the kind of methodologies that restrict innovation, but enough similarity so that we can share data across different mission types and durations and populations, etc. And that's where I think the value of the collaboration of everybody coming together to say, can we optimize methods so that there's a translational aspect to comparing data from different missions, different labs, different groups to the greatest extent possible. And so there's a lot of people working toward that. We hope we can pull that off. And then the other thing I wanted to add, just as kind of closing, is this research that we're doing is going to be so critical to the field, I think, to medicine on Earth, and also to the advancing of humans in space. One of the things that I think should always be at the center of our thinking is, how do we develop that into solutions in the shortest amount of time that is realistically possible? And that comes with a whole range of uncertainties, right? Because when do we know with so few flyers that it's the appropriate solution? But I think we should be very much solutions oriented in our thinking, so that as we're gathering this data, trying to generate meaning from it, that we're also thinking about the applied side of it. How can we apply this? And there's going to be tiers to that application. Some will be, yes, we know enough to act now. Some will be, we need more data. And some will be, we're quite far from being able to say we can execute on this. But let's think a lot about how we apply this in the safest way possible. So with that, I'll turn it back, and thank you, everybody. This has been a really fun day together.
So at this point, I'm going to have to apologize to people who did not have their questions addressed. We do store them, and we can distribute them to the speakers and email or post their responses, excuse me, to questions that we didn't get to. But I do want to turn it over to Chris Bradburne, because you play a role as the commentator, being well-versed in the science and the technology and operational aspects of this. But still kind of one half step removed by not yet being an investigator collaborating in human spaceflight projects. Hopefully we can change that soon. But from that perspective, I'm going to give you the last word.
Thanks, Mark. I think the thing I've got going for me is I'm a space nerd. So I really, I mean, I don't understand why people who aren't space nerds aren't, because this is just the coolest thing ever. And it's the edge of the frontier of exploration. And certainly what Chris Mason and Mike Schmidt and Mark Shelhamer, Malika, and others, are doing is super inspiring. And it's really cool to view this, seeing the confluence of all these fields: precision medicine, systems biology, the digital twin stuff, computational biology, sort of where I live, as well as nobody said anything about ML, AI, and all that kind of stuff. But that all definitely plays a role, right? Data science with huge compute. So I think all these things are going to come into play. The stuff that SpaceX is doing to make, human spaceflight accessible to not just hundreds, but thousands of people. Like, we're going to go beyond, like, once Starship is operational, it's going to be super interesting to see how many hundreds to thousands of people start flying. And then suddenly all these N of one, experiments and sort of the individual chronic effects that affect maybe 1 out of 1,000, 1 out of 100,000 or 1,000,000. Suddenly there's going to be enough people where you're going to see more of those things. So it's not just the performance, it's also going to be this health effects that all the research that you find folks are doing is going to hopefully be super impactful for. So thanks, Mark, for having me, and thanks to our speakers as well. I think we're 1 minute short.
Thank you. That's perfect timing for me. Thanks to everybody. First of all, I do want to say, even though I am wearing a shirt with a NASA logo on it, I do not speak for NASA, and I'm not representing necessarily the views of NASA or any potential government organization or any entity at all, except for myself. Now, I do want to wrap up and thank, obviously, Chris, and Michael, Malika as well, the Hopkins at Home people who put this on and make it look easy and seamless. It is not easy, but it is seamless because they do such a great job. And again, from the Office of Research and Translation, which supports this really behind the scenes by engaging with Hopkins at Home people and supporting some of my work. And that includes Larry Nagahara, who's the Vice Dean, and Linda McLean, who has done a lot of the logistical work for all of these things. I appreciate the efforts of all of these people and organizations. And of course, the audience, we would not do this without you and without feedback that we get from the community. So hopefully this is helpful. If you have any suggestions, ideas for topics that you might want to see covered in future mini symposia, please send them to me. No guarantees, but I will consider all reasonable suggestions. So with that, thanks again, everybody, and we'll see you on the next symposium.
Thanks so much.
Thank you.
Thanks, everyone.